Hello everyone, I am Dr. Sirajul Akfazi. I welcome you in my YouTube channel. Today in this video, I am going to talk about heterocyclic compounds. You know my friends, heterocyclic compounds play important role in chemistry. So learning of heterocyclic compound is important for us. So let us start learning of heterocyclic compounds. You know, heterocyclic compounds are organic compounds that contains ring composed of carbon and other atoms. Carbon and other atoms, heteroatoms, that is important. In natural heterocycles, mostly nitrogen, sulfur and oxygens. Nitrogen, sulfur and oxygens. Keep in your mind, nitrogen, sulfur and oxygen. Heterocycles exist as 3, 4, 5, 6 and multi-membered ring. Now question concerning to stability. Stability of heterocyclic compound increases with less distorted bond angles. Write it down. Less distorted bond angles means less strain that are in 5 and 6 member ring, and with maximum number of conjugated double bonds, because the delocalized pi bonding electron pair form a molecular pi orbitals filled with 6 electrons, and the compound have the aromatic character. Now you understand these 2 points. First point, less strain, and second point, maximum conjugations. Now my friends, some rings, in spite of being 5 or 6 member ring, are not stable heterocycles. Then take your pen and write it down. Cyclic hemiacetals. What? Cyclic hemiacetals. Cyclic forms of monosaccharides are in equilibria with their open acyclic form. Another example, cyclic ester, which is known as lactones, amides, which is known as lactam, and cyclic dicarboxylic acid anhydride can undergo hydrolytical splitting. Now, my friend, non-aromatic heterocycles, cyclic ether, amines and sulfides behaves like their acyclic analogues with the same functional group and do not require special discussions. Aromatic heterocycle with the maximum number of conjugated double bonds are much more important. Survey of the most important heterocycles. Look at here. Pyrrole, pyridine, imidazole, thiazole, thiophene, thiolane, furan, tetrahydrofuran, pyridine, piperidine, pyrimidines, pyrazines, azepine, 1,4-diazepine, 4-H-pyrene, tetrahydropyrene, indole, quinoline, isoquinoline, chromine and Pteridine. Now my friend, nomenclature of heterocycles. Common name for many ring systems are accepted by the IUPAC rules. Rings with maximum double bonds like furan, pyrrole, indole, pyridine accepted. Saturated ring system either prefixes dihydro, tetrahydro, pyrohydro, or in the 5-member ring, pyrroline, INE, pyrrolidine, IDINE. In 6-member ring, piperidine, piperazine. Look at here. Pyrrole, pyrroline, pyrrolidine, imidazole, imidazolidine, piperazine, and piperidine. Systematic name according to Hans Widman for monocyclic heterocycles. Prefixes indicating the number of heteroatoms. A stem of the same indicates the type of heteroatoms, ending for number of ring members and saturations. Some examples from rules used for some compound in biochemistry. You know number of heteroatoms, di, tri. Heteroatoms is thiox. Is thiox. Number of rings 1, 2, 1 is not possible. Here is the wrong. That is the 3 and 4. 5, 6, 7, 8. Unsaturation, irene, idine. Just correct it. It is not 1, it is 3. It is not 2, it is 4. Et, ole, ine, epene, osin. Saturated nitrogens and oxygenous sulfur. Irenes, erenes. Etidine, etane. Olidine, olane. Per, ene. Epane, epane. Okay. Now friends, nitrogen containing heterocycles are basic unless the unshared electrons pair of nitrogen is conjugated with pi electrons pair of double bonds. That is, unless it takes part on the formations of an aromatic 6 electrons alkaloids. You know my friend, pyridine is a weak base. The unshared electron pair of nitrogen atom can bind a proton by coordination to give a pyridinium cations. Pyrrole is not a base. The unshared electron pair of nitrogen is not able to add protons. Imidazole is a weak base, one of the 2 nitrogens you can see here. Now pyrrole is not a basic compound. The most important pyrrole derivatives are cyclic tetrapyrroles, heme, chlorophyll, and linear tetrapyrroles, bile pigments. You can see the cyclic tetrapyrroles, porphine and protoporphine and chlorine, and linear tetrapyrroles, and bilirubin, vitamin B12, cobalamin is essential for the maturations and development of red blood cells. Coenzyme B12, cobamide, cofactor 5 deoxy adenosyl cobalamin, and methylcobalamin. 5-deoxy adenosyl cobalamin. You can see the complex structure. Methylcobalamin, part of the molecules. Now, pyridine is a cyclic secondary amine, pKa 2.7. You can look at here the structure. Pyridine, it takes H plus and under equilibrium it will form pyridinium ion. Proline, you can see the pyrrolidine 2-carboxylic acid. You know proline is the only standard alpha amino acid that has a secondary amino group, and that is important point for NET, GATE, JAM examinations. Hydroxyproline. Proline hydroxylated in positions 4 or 3 is the characteristic constituents of collagens, the most abundant protein of connective tissue. 5-oxoproline, lactam of glutamic acid, is formed in the group translocations of amino acid gamma glutamyl. You can look at this to a structure of proline, 4-hydroxyproline, and pyroglutamate, 5-oxoproline. Now indole is not basic compound, 2,3-benzopyrrole. Now indole is not a basic compound. Look at here, tryptophan. The full form of tryptophan means complete IUPAC name 2-amino-3,3-indolyl propanoic acid is a standard amino acid from which biologically active compounds are derived. Look at the structure of tryptophan. There is a structure here which is known as benzopyrrole. Now serotonin, you know about the serotonin. It is 5-hydroxytryptamine. Alcoholic group is present at the 5 positions. Is a neurotransmitter in the central nervous system It has also acted as a vasoconstrictor released from the blood platelets. You know, melatonin, N-acetyl 5-methoxytryptamine, is released from the pineal glands and has a role in the control of diurnal biorhythms. You know, Indole 5,6 quinone that originate from the tyrosine is a melanogen. Now look at the structure. Hydroxy, methoxy, and indole 3,6 quinone. Now imidazole, you know, 1,3-diazole, commonly known. It is a weak base. One of the nitrogen atom can add proton, because you can see here 2 nitrogen, 1 nitrogen is able to additions of H plus ion, which is coming from the acid in the solvent. So imidazole in equilibrium with the imidazolium cation. Histidine, look at the structure of histidine, which contains imidazole. It is a part of amino acid. Histidine 2-amino 3,4-imidazolyl and propanic acid is an essential amino acid. The side chain of the histidyl residue are responsible for the buffering ability of protein at a physiological pH values. Because only the imidazolyl groups can take part in the proteolytic reactions under those conditions. You know about the histamine originates by decarboxylation of histidine and act as a vasodilator in inflammations and allergic reactions. It also causes contractions of the smooth muscles of the bronchial stems and the acidic stomach secretions. Look at the structure of histamine. Now I am going to talk about thiazole. There is a sulfur and nitrogen in 5-member ring at 1,3 position. So thiazole ring occurs in thiamine, vitamin B1, and that also contains a pyrimidine ring. Look at here the thiazole. Thiamine diphosphate is the coenzyme that transport active aldehyde groups in the course of the oxidative decarboxylations of pyruvate and other alpha-keto-carboxylic acid. The fully saturated thiazolidine tetrahydrothiazoles ring appears in the structure of antibiotics penicillins, and the effect of which is caused by the unusual beta-lactam ring. Look at this structure of beta-lactam. Now thiolane is completely hydrogenated thiophene. This ring structure appears fused with the imidazolidone in biotin. You can look at the structure thiophene and look at the structure of thiolane after dehydrogenations. In the cells, biotin may bind carbon dioxide and resulting carboxybiotin serves as a donor of carboxyl in the important carboxylations. Biosynthesis of fatty acid, synthesis of oxalactoacetate from the pyruvate. 1,3-dithionylane is a cyclic disulfide that can be easily reduced to a cyclic alkane 1,4-dithiols. It occurs as a part of lipoic acid. Look at the structure. Now oxygen containing 5 and 6-membered heterocycles. Furan derivatives are rather rare in the natural compounds. On the contrary, tetrahydrofuran ring is quite common in cyclic forms of sugars. So deribofuranose contains tetrahydrofuran. Look at the furan structure. Pyridine is a labile ring without the aromatic electron sextet, but it is stabilized through oxidation to pyrone. Tetrahydropyrene ring occurs in cyclic form of monosaccharide. So 4H pyrene and 4H pyranone, gamma pyrone. Many plant substances comprise the pyrene ring that is usually fused condensed with the benzene ring. Look at the chromane and chromone. Chromane and chromone. Those structure appears examples in the flavonoids in the active components of heme tetrahydrocannabinol THC and tocopherols and derivatives of coumarin. Tocopherols are important exogenous lipophilic antioxidants which prevent oxidations of membrane lipids and lipoprotein by reactive oxygen species. Look at here alpha tocopherol, which is known as vitamin E. The complete name of tocopherol or alpha tocopherol is 578 trimethyl derivative of the phenol tocol. Now derivatives of coumarin. Warfarin, biscoumarins prevent in vivo coagulations of blood. They act as antivitamins K by inhibitions of biosynthesis of vitamin K dependent coagulations factor. Now my friend, pyridine is a weak base. pKb is equal to 8.7. You look at here the structure which is containing nitrogen with the 5 carbon pyridine. It takes protons from acid and forming pyridinium. And the most important derivative of pyridine are two essential factors, nicotinic acid and nicotinamide is called niacin or pellagra preventing factor PPF, and they are included in the group of vitamin B. Look at here nicotinic acid structure, which is known as pyridine 3 carboxylic acid, and nicotinamide, which is present in the alkaloid nicotine. Now look at the vitamin B6 is a group of name for 3 related derivatives of pyridoxine, pyridoxal and pyridoxamine. Look at the structure of pyridoxamine, pyridoxal and pyridoxine. Now NAD plus. NAD plus means what? Nicotinamide adenine dinucleotide, and NADP plus NAD phosphate are coenzymes of dehydrogenase. Look at the structure. Anhydride bond is there, adenine groups are there, sugar groups are there. The active group of NAD plus as well as of similar NAD plus is nicotinamide. It acts as an oxidant that takes off 2 atoms of hydrogen from the substrate. 1 atom plus 1 electrons, like hydride anion, is added to the para position of the pyridinium ring. The remaining H plus bind to the enzyme. Look at here H plus and H minus. Oxidized form of NAD plus. Reduced form of NADH plus H plus. So quinonoid ring, no charge. You know my friend, pyridoxal phosphate. Look at the structure. Pyridoxine, generally known as vitamin B6, and pyridoxal phosphate group. You know the carbon and oxygen bond and phosphate group, this bond is highly energetic. So pyridoxal phosphate is the prosthetic group in many enzymes taking significant part in the alpha amino acid metabolism, namely. My friend, they catalyze transaminations and decarboxylation of amino acid. The aldehyde group of pyridoxal coenzyme bind to a alpha amino groups of an amino acid, forming an LD means intermediate, which is known as a ship base. My friends, pyrimidine is a dibasic compounds. Look at the Widman nomenclature. 1,3-diazine. 1,3-diazine is known as pyrimidine. 3 pyrimidines bases are components of nucleic acid: cytosine, uracil and thymine. Cytosine, uracil and thymine. Cytosine is known as 4-amino 2-hydroxypyrimidine, uracil 2,4-dihydroxypyrimidine, and thymine 5-methyl-uracil. And they exist in 2 tautomeric forms. Only that lactam forms can be ribosylated to give the nucleosides. You can see the lactam form and lactam form of uracil. So pyrimidine ring is comprised also in the structure of thymine and barbituric acid. My friends, look at the purines. Pyrimidine imidazoles. Pyrimidinoimidazoles. A typical numbering, you can see the starting here is different from the what concept we have learned in the general chemistry. Here starting is nitrogen 1, which is not containing hydrogens. So starting nitrogen 1, 2, nitrogen 3, 4 and 5, 6, 7, 8, 9. Breaching or fused to carbon is also numbered accordingly. So 2 purine bases of nucleic acid, adenine and guanine, 6 amino purines and 2 amino 6 hydroxypurine, hypoxanthine and xanthine, 6 hydroxypurines and 2 6 dihydroxypurines. Now look at the uric acid, also the same structure present in uric acid, 2 6 8 trihydroxypurine. There are 3 hydroxyl groups in the purine ring, and lactone form of uric acid. Now friend, methylxanthines are N-methyl derivative of xanthines, known alkaloids in the coffee, tea and cocoa. Look at the structure. All structure contains the same ring, only substituent is different. On the basis of that, there is a nomenclature is different. Caffeine 137 trimethyl xanthine, theophylline 1,3-dimethyl xanthine, and theobromine 1,7-dimethyl xanthine. So allopurinol is an isomer of hypoxanthine, which is used in treatment of hyperuric acidemia and gout. It inhibits xanthine oxidase, the enzyme that transforms hypoxanthines and xanthine to urates. Now look at the mercaptopurines, which contain the sulfur. 6 sulfuri purines inhibit the biosynthesis of purines bases, and it is used in the chemotherapy of cancer. These 2 compounds, which contains allopurinol and mercaptopurine. Now Pteridine, Pyrimidine, Pyridines. So again, numbering scheme is very different here. Nitrogen got 1, another nitrogen 3 and 5 and 8. So 3 important coenzymes are derived from the pteridine and coenzyme folic acid and flavin coenzymes. In the cell, the pteridine system originates from guanine. Now friend, BH4. What is the BH4? Tetrahydrobiopterin act as reducing coenzyme in the certain hydroxylations catalyzed by monooxygenase. It supplies 2 atoms of hydrogen is to oxidize to the quinonoid form of dihydropterin. So look at the tetrahydrobiopterin, which is known as BH4. Why it is BH4? Because 4 hydrogen will transfer from here, and then after removing 2 hydrogen, oxidize quinonoid form. Now folic acid. What is the meaning of folic acid or folate? It is a N-pteroglutamic acid. The number of attached glutamyl residue may be higher, although synthesizing the pteridine ring, mammals are unable to conjugate it with the PABA and glutamate. They have to obtain folate from their diets or from the microorganism in their intestines. Look at the pteroic acid structure. The ring is there, 2 amino, 4 hydroxy pteridine, CH2 and para-amino benzoate, glutamate, N is equal to here 1 to 5. Now tetrahydrofolate, H4 folate is a cofactor that transport 1 carbon groups, aldehyde, alcohol and CH3, inevitable namely for the biosynthesis of purine bases and thymine. Look at the site for bonding of 1 carbon atom. Methoxytetraxase 4 amino termethyl folate and aminopteridine 4 amino folate are antifolates drug used as a cytostatics in the oncology. Now friends, very interesting structure here which contains the ring, which is heterocyclic. By condensation with the benzyl ring, pteridine gives benzopteridine, the 2,4-dioxyderivative of which is isoalloxazine. Look at the structure of isoalloxazine, where introductions of benzene ring occurs in the heterocycles. This is known as isoalloxanes, which is known as also benzene G-pteridine. What is the meaning of here? Brackets small g, it is the fused ring with the benzenes. Now look at the structure of riboflavin. The same structure here, also benzene ring attached with the purines. This is known as vitamin B2. Is 6,7-dimethyl 9,1-ribityl isoalloxanes. So ribityl is the remainder of the sugar alcohol ribitol. Riboflavin is only colored hydrophilic ethylamine intensive yellow. So it is a part of coenzyme FAD and FMN. My friends, FAD means what? FAD is flavin adenine dinucleotide and FMN means flavin mononucleotides. So look at the structure of coenzyme FAD oxidized form. Here you have the sugar, you have purine bases, you have the 2-phosphate, and again the structure alloxazines. Flavin prosthetic groups act as oxidant in certain types of reaction catalyzed by dehydrogenases or oxidases. Look at the oxidized form and reduced form. Now my friends, the heterocycles is in major psychotherapeutic agents. For example, thiazine, phenothiazine, chloropromazines. Look at the structure of chloropromazines, tranquilizers, neuroleptics and psychostatics, azepine, thiopine, antiazepines, antidepressant, thymoleptics and psychostimulants. So look at the structure where contains azepine, 7-member heterocyclic compounds. Now anxiolytics. Anxiolytics is the containing, you see the benzo diazepines, and diazepine which is known as Valium. Some other derivatives of benzodiazepam are hypnotics and fluotazepines, arifonols or myorelaxantins. So my friends, now you know the alkaloids. Alkaloids are natural nitrogenous bases, originate from amino acid, sometimes called secondary metabolites. Occurs mostly in higher plants. Most of them are heterocyclic compounds. They exhibit extraordinary variety of structural features. Many of them have a marked biological effects. Quite a number of them possess curative properties. My friend, it is not yet known whether alkaloids are unwanted byproducts or products of the detoxification, protective or reserve compounds or useful regulators. So alkaloids are weak bases. You can see the alkaloids and pKa value: atropine, ephedrine, quinine, cocaine, nicotine, morphine, papaverine, caffeine. So all are alkaloids with the different pKa value. And free alkaloid bases converted into salt which is soluble in water. Now example of basic alkaloid like nicotine, vasoconstrictive stimulants, quinine, antimalarial, anti-arrhythmic quinidine, morphine, narcotic actions. Now papaverine, anti-spasmodic action. So now look at here, tropine and atropine, anti-spasmodic actions, cocaine, local anesthetic, psychostimulants, and lysergic acid also contains the heterocyclic ring, and methyl lergometidine, uterolotonic. So all compounds contains heterocyclic compound. So my friends, I hope this video will be helpful to you to understand the importance of heterocyclic compounds. If you like this video, then subscribe my channel, and thanks for watching.